the brain and behavior, the structure of the brain. Um, my hair's all messed up. I was just outside. I can smell a wood fire, and nobody around here burns wood, so I was afraid something was on fire. I went out to the barn to make sure my barn was okay. Found three raccoons in there. At least my dog did. They went running all over the place. He didn't catch anybody. That's a good thing. So let's go ahead and get started. Lifespan development of the brain and behavior. Chapter 7. The brain contains about 100 billion neurons and an equal number of glial cells. It is estimated that there may be as many as 100 trillion connections within the brain. While genetics plays an important role in creating the brilliant creatures that each and every one of us may be, the environment guides the process of development. The most rapid brain growth is during gestation, but it continues to expand markedly through the language acquisition years. For an egg to be fertilized and come to term, everything has to be perfect. The sperm must have its full complement of 23 chromosomes and will not be able to swim through the cervix, up through the uterus, and into the fallopian tube to penetrate the ova unless it is perfect. The ova must have its full complement of 23 chromosomes and be in the right position when the sperm reaches it. For implantation to take place, the uterine wall must be ready for implantation. The zygote must implant near the top of the uterus. Adequate vascularization must take place between the embryo and the uterus. Growth and cell division must take place in a continual and balanced manner. As soon as the sperm penetrates the ova, the structure becomes a zygote. Within 12 hours, the single cell divides into two cells. Within 2 weeks, the zygote becomes an embryo and divides into three distinct layers. The nervous system develops from the outer layer of the three layers, the ectoderm. The ectoderm grows into a flat oval plate. The cells of the plate do not grow at the same rate, and a groove develops. This groove is known as the primitive streak, and that's the primitive streak right there. As the cells continue to divide, the groove slowly grows into the neural tube. The front or anterior portion of the tube divides into three structures that will become the hindbrain, the midbrain, and the forebrain. By the 8th week of gestation, the fetus has developed all the rudimentary organs in its body. The brain at this time makes up 1/2 of the fetus's mass. The brain will continue to grow through the teenage years, actually into the age 25. Nervous system development takes place in 6 stages: neurogenesis, the formation of neurons; cell migration, movement of neurons to form nerve cell populations; differentiation, development of distinctive types of neurons; synaptogenesis, development of synaptic connections as axons and dendrites grow; neuronal cell death, selective death of neurons; synapse rearrangement, refinement of synaptic connections. The first stage of nerve system development is neurogenesis. This is when the nerve cells are produced. Nerve cells themselves do not divide, but the pre-nerve cells, which are located in the inner layer of the neural tube, do divide and create a closely packed layer of cells called the ventricular zone. And this is what the ventricular zone looks like. The cells of the ventricular zone continue to divide and give rise to daughter cells, which also divide. All the body's neurons and glial cells are derived from ventricular mitosis. Neural cells will be completely developed by birth. Each neural structure in the brain will develop at the same time in gestation for all humans. Neurons of the developing nervous system are always on the move. During the cell migration stage, the cells of the ventricular layer begin to move where they will end up. In humans and other primates, by birth, All the neuronal cells will have found their way to where they will always be. Some neurons will creep down glial cells known as radial glial cells. Neurons can either move down the glial cells or jump from one to the next. This migration is guided by various chemicals called cell adhesion molecules. The cerebrum is formed by wave after wave of neurons till the entire cerebral cortex is formed. Once the pre-neuronal cells reach their destination, genes in the cells begin to make the proteins that are required by neurons. At this point, the neurons begin differentiating into the distinctive neurons of the region that they are in. Most neuronal cells differentiate through induction. They take on the task of all their neighboring cells. Some cells are undifferentiated cells and will differentiate into any cells that they are close to. And these are known as stem cells. So stem cell research can be huge, but it's against the law in the United States. It was a law signed by George W. Bush back in 2008, I believe. When neurons migrate, well, actually what happened was they passed the stem cell research bill, and it passed through Congress, and he vetoed it, making it something that we don't do in the United States. When neurons migrate to their final destination, they start the process of synaptogenesis. Axons and dendrites begin to web out, making contact with the cells that they are going to be responding to. This is done through swollen ends of dendrites and axons called growth cones. The growth cones reach out using fine filaments, known as filopodia, or plates, known as lamellipodia. Growth cones are drawn by chemical signals called chemoattractants. Growth cones can also be repelled by chemical signals called chemorepellents. As adults, we maintain synaptogenesis structures, dendritic growth cones, axonic chemoattractants, and chemorepellents. Synapses can form rapidly on dendrites and dendritic spines. The number of spines increase rapidly after birth and are affected by experience. Neuronal cell death, apoptosis, is crucial to brain development, especially during the embryonic stages. Neuronal cell death ranges from 20 to 80%, varying from region to region of the brain and spinal cord. An area needs only so many neurons and synapses, and as the area grows, more neurons than are needed flood into the area to ensure that the most perfect neuronal structure and synapse are created. The cells that die are self-selecting to die. They are committing suicide. All cells carry a death gene that causes a sudden influx and release of calcium ions that causes the mitochondria to release a protein called Diablo, which is Spanish, of course, for the devil. Diablo binds the inhibitors of apoptosis proteins, the IAPs, that have been inhibiting a family of proteins called caspases. The caspases are proteases, or enzymes that dissolve protein. The caspases break down the protein and the DNA of the neuron. Normally, Diablo is inhibited by BCL2. Cells that are able to make proper and adequate synaptic connections are the ones that live, while those that don't die rather than make a poor connection. Chemicals that enable proper growth are called neurotropic factors. And this is a joke, integrating signals hinting at obsolescence. George undergoes apoptosis and his brain explodes. Scientists have discovered a substance that promotes the growth of spinal ganglia that they call nerve growth factor. If a technique could be developed to use NGF effectively, spinal injuries could be repaired back to normal function. With time and experience, some synapses disengage and other synapses are formed. This process is especially prevalent during cell death in the area, refining the remaining synapses to provide optimum connections. While neurons and glial cells develop from the same source cells, scientists don't know what informs the cells to end up as one or the other. While neuronal growth takes place almost exclusively before birth, glial cells have their greatest growth surge right after birth and continue to grow throughout life. The glial cells provide myelin for the axons of the neuron. The myelin protects, feeds, and accelerates the electrical response to the neuron. Myelination allows people to walk with coordination and the brain to process information rapidly. Multiple sclerosis is an autoimmune disease that affects select individuals. The condition allows the immune system to attack the myelin of the neurons and leave gaps in the structure that slows the neuronal response. Intrinsic factors in the development of the nervous system deal with genetic factors that allow for proper development. When genetic aberrations occur, it can cause abnormal brain development. Tay-Sachs disease causes the destruction of neurons that eventually results in death. It has been identified as a flaw on the 15th chromosome. And of course, Tay-Sachs disease is a disease that we have seen only in the Ashkenazi Jews, the Jewish population that has lived in Eastern Europe. Jewish people tend to marry within their own population, and because of that, it has created there was a mutation, Tay-Sachs disease, mutation on the 15th chromosome. And because of the intermarriage that takes place in that population, it has repeated itself, and so it is only in that population. Down syndrome causes intellectual disability and body abnormalities. It has been identified as an extra chromosome on the 21st pair. Extrinsic factors include malnutrition, fetal alcohol syndrome, hypoxia-induced intellectual disability. Hypoxia is not enough oxygen. Usually that's at birth. The baby starts to breathe before it has cleared the birth canal. The sum of the intrinsic, or it has the umbilical cord has wrapped around the baby's neck, and it's shut off the blood that normally gives it oxygen as it's passing through the birth canal, hypoxia. The sum of the intrinsic factors are referred to as the genotype. The genotype plus the extrinsic factors represent an individual's phenotype. And we used to deal with this when I was working in the lab. When you're doing a cross-matching blood, you've got a donor's blood that you're going to give a pint of blood to the recipient, and you have to take the recipient's blood and make sure that the donor's blood will not cause a problem. So what you look at is, of course, you see the phenotype. The phenotype is the O positive or whatever, but from time to time they will have a genotype that is incompatible with the recipient's blood. So what we have to do is check the genotype to make sure that it won't interfere with the patient's blood. And that's what a cross match is. We are cross matching the blood to make sure that it is compatible with between the donor and the recipient. The phenotype is what we see, the O positive. The genotype is any hidden factors in the blood. When mutations take place in a species' genetic history, the mutation must often create a maladaptive circumstance. But on the rare occasion when the mutation is actually useful to the species, the mutation will be reproduced in the offspring. Experience is an important factor in brain development. The human brain is only 1/4 its adult size at birth, yet few new neurons are added. The reason for the exceptional growth is due to dendrite growth and myelination. Dendrite growth and myelination are induced by experience with the various muscles and sensory organs of the body. One form of extrinsic stimulation causing a problem is amblyopia, also called walleye and lazy eye. Children with the problem have a misalignment of the balance of their binocular vision. If left untreated, by age 7 or 8, the suppressed eye will be totally blind. And this happened to my niece, great niece. She's my great niece. Her mother put her, her mother didn't want to pay a lot of attention to her, so she would turn on the television, lay her down on the Davenport, on the couch, and give her a bottle. And so her right eye was, she laid her on her right side, and so her right eye was covered, and only her left eye could see the television. So here she's got a bottle in front of her, she's drinking her milk, and her right eye is covered. And what she did was develop amblyopia in that eye because of, this was a continual process that the mother did. And it gave her walleye. And to this day, she still has walleye. She almost blinded her daughter, my niece. While treatment in childhood will result in perfect vision, when the problem is corrected in adulthood, the eye does not gain acute vision. And this is what it looks like. This is because when the problem is allowed to remain the same over time, the neural connections in the brain from the weak eye are not as intricate. Understanding of amblyopia and other asymmetrically balanced neural connections has been studied by performing binocular deprivation research on laboratory animals. And of course, this is how you cure walleye, is by covering one of the eyes. And you cover the strong eye and force the weak eye to form connections. That's how amblyopia is cured. Researchers have discovered that sensory organs have a sensitive period when the neural development is crucial for stimulation to induce proper dendritic connections. If stimulation does not occur by this time, recovery to a normal state is impossible. To understand why people tend to have a dominant eye, it must be remembered that each eye represents millions of receptors vying for attention in the brain. When one eye receives more stimulation than the other, some of the synapses in the brain connected to the unstimulated eye become weaker. While most synapses do not fluctuate in their strength with stimulation, some do. These synapses are known as Hebbian synapses. 75 years ago, the leading cause of intellectual disability in the United States was phenylketonuria. 2% of the population carries the recessive gene. The gene controls the enzyme that breaks down phenylalanine, an amino acid in protein. Because the enzyme does not break down the phenylalanine, a toxic level collects in the brain and destroys brain cells, and this is known as phenylketonuria. Urea is a waste product. So this is the waste product of phenylketones. Actually, ketone is a waste product as well. It's the waste product of the phenylalanine. And I used to draw these PKUs from newborn babies. That was one of the things you had to do with a newborn. You had to draw a PKU from them to find out if they had phenylketonuria. Williams syndrome is a relatively rare genetic abnormality that causes neural and facial abnormalities as well as mild intellectual disability. Research indicates that Williams syndrome is caused by an incomplete chromosomal structure on the 7th chromosome. These individuals have normal linguistic ability but difficulty in learning by observing. Individuals with Williams syndrome have very characteristic facial features. They have broad foreheads, small eye openings. They have low nasal bridges, nostrils that point forward, a long area between the nose and the upper lip, full cheeks, large downturned mouth. They all look alike. as weird as that seems. One of the interesting things about Williams syndrome, however, is that they are really happy people. They they don't feel unhappy. They always feel happy. It's really strange. Down syndrome is a condition caused by the addition of an extra chromosome among the 21st pair of chromosomes. This abnormality can cause mild to severe intellectual disability and various physical anomalies, heart malformations, and brittle arteries that lead to a shortened life expectancy. And there you go, 21st pair. And this is a female. These are X chromosomes. This is the 23rd pair. Trisomy 21 is more prevalent in older women, probably because of the age of the ova. Research shows that individuals with trisomy 21 have abnormal formations of their dendritic spines, making it more difficult to learn. The most frequent form of inherited intellectual disability today is Fragile X syndrome. The DNA of this select chromosome seems more pinched and fragile, more likely to break off. And there it is right there. That is the chunk that's about to break off. The real problem seems to be an excessively repeated trinucleotide that is in an abundance 4 times normal, thus causing the extended appearance of the chromosome. And you can see it right there. This is a female. Genetic normal structure, so the head is normal. The forehead is broad. They have elongated faces, large prominent ears. They have crossed eyes or strabismus, highly arched palate. They have hyperextensible joints. They have hand calluses from self-abuse, from rubbing their hands together. They have pectus excavatum, or an indentation in their chest. They have mitral valve prolapse, which is a benign heart condition. They have enlarged testicles if they are a male. They have hypotonia, which is low muscle tone. They have soft, fleshy skin, flat feet, and about 10% of them will have epilepsy. About 40% of children born to alcoholic mothers show a distinctive profile of anatomical, physiological, and behavioral impairments known as fetal alcohol syndrome. Children suffering from fetal alcohol syndrome show stunted growth and select facial abnormalities, small brain, small eye sockets, flat midface, indistinct philtrum, that's the groove under your nose to your upper lip. They have a thin upper lip. They have small chins, short noses, lowered ears, low nasal bridge, and epicanthic fold in the eyelids. Epicanthic fold is a layer of fat in the eyelid. Brain impairment is due to their small brain and brain structure. Problems like the almost absent corpus callosum, that's the connection between the hemispheres. Here's a normal corpus callosum, and here is a fetal alcohol syndrome individual. As you can see, the corpus callosum is almost absent. Reduction of cerebral cortical gyri, the folds in the brain that gives it more surface. And you can see this is a normal baby's brain, and this is one with a child with fetal alcohol syndrome. You can see that there are far fewer gyri in this brain than there is in that brain. Intellectual disability can be mild to severe, possibly depending on the time during pregnancy and the level of consumption. Besides intellectual disability, children with FAS also show such neurological abnormalities as hyperactivity, irritability, and tremulousness. Studies also seem to indicate that marijuana may have the same effect as alcohol. What were we going to talk about? The type of drinking that seems to be the most dangerous for a baby is binge drinking, where an individual will abstain through the week and then they'll get drunk over the weekend, drink very heavily at one time. Autism spectrum disorder is a lifelong developmental disorder that is characterized by slight to severe social impairment in language. 70% of children with ASD develop poor language skills, rarely getting beyond monosyllabic responses and echolalia. When I first started studying psychology, um, uh, autism they said that autism had 70% mutism rate. And now that we have learned more about autism, we are better at training individuals who have autism to speak. Uh, Autism spectrum disorder doesn't have to correspond with any mental deficiency, but the lack of social interaction may impair the diagnosis. Normally, when an individual meets a stranger, they scan their faces for recognition and potentially put this information in their long term memory. However, individuals with autism show brain scans where they seek no recognition and therefore have a difficult time making new acquaintances. Alcohol spectrum disorder seems to have something to do with brain organization. Practically all information is organized differently from a normal control. Alcohol spectrum autism I'm sorry autism seems to affect from 1 to 2 children per thousand. It is far more common among male children than female children and seems to run in families. And I just saw figures that were a lot stronger than that. Well, yeah, okay, it's 1 in 37 now. Various areas of the brain show abnormalities among autism spectrum disorder children, including the corpus callosum. Autism spectrum disorder, formerly called Asperger's syndrome, seems to be a less severe form of autism spectrum disorder, where the individual does not suffer from language deficits but has problems with social interactions. As people age, there seems to be a steady decline in brain size that begins as early as the 30s and begins to accelerate after age 45. However, the degree of decline seems to vary from individual to individual, from barely evident to exaggerated. Yet, brain expansion seems to continue to occur, as is evidenced by the presence of growth cones in the frontal lobe, even in the oldest individual. As an individual enters their fifth decade, their hippocampal formation begins to shrink. The supratemporal temporal gyrus also loses volume. In fact, most areas of the brain begin to lose volume. Over 4 million Americans suffer from Alzheimer's disease. Oddly, the possibility of developing symptoms of Alzheimer's increases with age until the age of 85 and then starts to decline for those people who have never developed symptoms. Alzheimer's disease starts as memory loss but progresses into greater and greater cognitive function decline until the individual can no longer carry on a conversation. Alzheimer's is accompanied by a marked cortical atrophy, especially in the frontal, temporal, and parietal areas. The brains of Alzheimer's patients show denigration of axon terminals and dendrites caused by the buildup of beta amyloid forming senile plaque. Amyloid precursor protein is bound by two enzymes, beta secretase and presenilin. If one of these enzymes mutates, amyloid plaque builds up. Some cells show abnormalities called neurofibrillary tangles. These are tangles of the neurofilament that are produced in abundance in the presence of the protein tau. Another gene mutation may allow Alzheimer's disease. APOE4 is supposed to break down the amyloid plaque, but is less efficient than the APOE2 or APOE3 versions. With both amyloid plaque and neurofibrillary tangles clogging the former functional neurons of the brain, the basal forebrain nuclei die, the cells that produce acetylcholine, and the memory function of the brain dies with them. And here we go. So let's take a quick look. Okay.
Well, my memory, I think, is good. I think it's good. But, I mean, I know telephone numbers and my car and everything else. But sometimes it gets blurry.
My memory? I think it's pretty good. But important things I don't seem to remember. If I could, I would be out of college.
I was in denial about how bad the Alzheimer's was, but I saw it progressing. It was getting worse. He was sleeping more and more. He was just like out of it sometimes. My dad was able to do everything, and now he's able to barely care for himself.
William and Harvey are just two of the 5.3 million people in the United States living with Alzheimer's disease, according to the Alzheimer's Association, and a new case is diagnosed every 70 seconds.
Alzheimer's disease is what we call a progressive neurodegenerative disease, and that's just a fancy way of saying that cells in the brain, or neurons, are dying because of the disease process.
While no one really knows what causes Alzheimer's, some of the signs include impaired memory, restlessness, language deterioration, emotional apathy, impaired behavior, and confusion.
And the family is great. We had 2 sons, and one was a PhD, and I can't remember the other one.
My father, he used to own a gin mill. Then he went into the butcher shop business, and he also delivered beer and soda around, and there were people working for him. It was an interesting life. He made it interesting, so did my mother. And they're still both around, too. Now I'm 92 years old. How about that?
If memory changes lead to real functional problems, people are not recognizing people that they've known for years. People are forgetting how to get to places they've gone to for years. Or people just aren't able to remember things that they used to be able to remember, to the point that it's affecting their ability to live their life, to carry out their daily tasks. That's when it's time to seek the help of a professional.
That's when Linda decided she needed some assistance.
People would come to the house and he would just let them in. Strangers, perfect strangers, he would let into the house. And they would stay for hours. And I would worry that they'd be walking around my house, checking it out for later, or possibly taking things, or I didn't know, he was signing contracts for services that we didn't need.
Now Linda takes Harvey to a special day center for Alzheimer's patients, where he spends time with his new friend William and a dozen other patients. Both men are living more stimulating lives as the day center helps them to adjust to their future.
Ta-da! Okay, let's watch this.
Feeling my husband at 8 inches after this honey hack was like the first time I was soaked like a waterfall.
Try this honey trick.
The human brain is a remarkable organ. Complex chemical and electrical processes take place within our brains that let us speak, move, see, remember, feel emotions, and make decisions. Inside a normal, healthy brain, billions of cells, called neurons, constantly communicate with one another. They receive messages from each other as electrical charges travel down the axon to the end of the neuron. The electrical charges release chemical messengers called neurotransmitters. The transmitters move across microscopic gaps or synapses between neurons. They bind to receptor sites on the dendrites of the next neuron. This cellular circuitry enables communication within the brain. Healthy neurotransmission is important for the brain to function well. Alzheimer's disease disrupts this intricate interplay. By compromising the ability of neurons to communicate with one another, the disease over time destroys memory and thinking skills. Scientific research has revealed some of the brain changes that take place in Alzheimer's disease. Abnormal structures called beta amyloid plaques and neurofibrillary tangles are classic biological hallmarks of the disease. Plaques form when specific proteins in the neuron's cell membrane are processed differently. Normally, an enzyme called alpha secretase snips amyloid precursor protein, or APP, releasing a fragment. A second enzyme, gamma secretase, also snips APP in another place. These released fragments are thought to benefit neurons. In Alzheimer's disease, the first cut is made most often by another enzyme, beta secretase. That, combined with the cut made by gamma secretase, results in the release of short fragments of APP called beta amyloid. When these fragments clump together, they become toxic and interfere with the function of neurons. As more fragments are added, these oligomers increase in size and become insoluble, eventually forming beta amyloid plaques. Neurofibrillary tangles are made when a protein called tau is modified. In normal brain cells, tau stabilizes structures critical to the cell's internal transport system. Nutrients and other cellular cargo are carried up and down the structures called microtubules to all parts of the neuron. In Alzheimer's disease, abnormal tau separates from the microtubules, causing them to fall apart. Strands of this tau combine to form tangles inside the neuron, disabling the transport system and destroying the cell. Neurons in certain brain regions disconnect from each other and eventually die, causing memory loss. As these processes continue, the brain shrinks and loses function. We now know a great deal about changes that take place in the brain with Alzheimer's disease, but there is still much to learn. What other changes are taking place in the aging brain and its cells? And what influence do other diseases, genetics, and lifestyle factors have on the risk of developing Alzheimer's disease as the brain and body age? Scientific research is helping to unravel the mystery of Alzheimer's and related brain disorders. As we learn more, researchers move ever closer to discovering ways to treat and ultimately prevent this devastating fatal disease.
The first time I ever did an autopsy, of course, I was just assisting. The pathologist had us remove the brain, and the brain fits pretty well inside somebody's skull. It's pretty big, but we took out that brain, and the guy had Alzheimer's disease, and it was about half the size it should have been. It was really, really tiny. Anyway, so I'll talk to you guys next week.